Well, first I'd like to thank the organizer and especially Shripad Donavali for inviting me here. It's my first time in India, so it's very exciting. And for me, it's particularly exciting because I've been working with Shripad for about 5 years now. And for me, it makes perfect sense to finally be here and in the very place that we've been doing this work. So what I'm going to talk about today is something I call next generation drug repositioning. So thanks, Pan, for this amazing introduction. That's perfect. You gave all the details before I can give my talk. So the idea is to use high throughput screening technology to improve cancer treatment. But first of all, the session here is called Why Metronomics, so I felt like I had somehow to answer this question. The word metronomics has been around for about 10 years since the invention of metronomic chemotherapy with the work of Bob Cabell and Judah Folkman in 2000. And people have been using it on and off. And Nicola, Shripad, and myself, when we wrote a piece 3 years ago for the Lancet Oncology, we proposed to use that generic term to design, to specifically address all the treatments that were using metronomic chemotherapy with drug repurposing, because in the clinic it's quite common. So with the metronomic chemotherapy, you're going to target the tumor vasculature and the immune system and the cancer cells to a lesser extent. And with the drug repositioning, you can really target cancer cells with new targets and also tumor vasculature and the immune system. So before I go into what I call next generation drug repositioning, I wanted to talk to you a little bit about beta blockers and what we've been doing in this field. So really what sparked everything for us, and when I say us, it's myself and Nicola Andre, because we work together on all these projects, is this paper that came out in the New England Journal of Medicine in 2008, and which reported something that's quite rare. It's a completely serendipitous discovery, that propranolol was very effective in treating severe hemangiomas. So for those of you who don't know hemangiomas, it's a very common benign vascular tumors that affects 2 to 5% of newborns. In most cases, it will regress by itself, but sometimes it doesn't, in about 15% of cases. And in this instance, it can be life-threatening depending on where the angiosarcoma is located. And up until this paper came out, there was not really any standard of care for these patients. A lot of people use high-dose corticoids, but didn't really work so well. Some others used interferon and even chemotherapy with incristin. And what happened in this hospital, they had patients like this patient that had like a really big hemangioma that was not going away, although he was on 4 weeks of high-dose corticoids. And this particular patient started to develop hypertension as a result of the corticoid treatments. So they had to give him an anti-hypertensive treatment. And after 1 week with propranolol, they've noticed that there was a whitening of the hemangioma, an opening of the eye, and a decrease in the swelling. So then they took a gamble. They thought, okay, this must be the propranolol effect. So they stopped the corticoids and just kept going with the propranolol. And this is what happens after 7 months of propranolol treatment. So in this study, there was 13 patients. Every single one of them responded completely. And it has completely revolutionized the treatment of hemangiomas around the world. So when we saw this paper, Nicola and I thought maybe we could use beta blockers in cancer treatment. And the hypothesis we came up with was, if we were to combine beta blockers with chemotherapy, maybe we could increase the efficacy of chemotherapy, because these drugs are clearly anti-angiogenic and they might have as well effect on cancer cells. So my background is angiogenesis and tumor angiogenesis, and that's why Bob Cabell is my absolute idol. And so what I use in the lab is I use the matrigel assay to basically, and that's when the thing is not working. Okay, no worries. So with the angiogenesis assay, you can quickly screen for new drugs and new drug combinations to see if they are anti-angiogenic. So what I did when I went back to my lab was to test whether hypothesis was correct. So what you can see here is if we use a dose of propranolol that doesn't have any effect on angiogenesis by itself, if we combine it with, in this instance, it's paclitaxel, but we can do it with other chemo drugs, it increases the anti-angiogenic effect of the chemotherapy. So as I was doing these experiments, retrospective analysis started coming out, first in OncoTarget and then in the Journal of Clinical Oncology. And they all showed the same thing. When we look back at clinical data from breast cancer patients, if they were treated for hypertension with beta blockers, somehow they're doing better than the other patients in terms of long-term survival, or relapse, and even metastasis. So because there was all this data on breast cancer, that's the first model we started working on. And using a triple-negative breast cancer model, we're able to show that the addition of propranolol to paclitaxel was significantly increasing the survival of the mice. And so we published this in 2011 in OncoTarget. And then we did a much larger study in neuroblastoma where we used 7 different beta blockers with 10 different chemo drugs that are used in the clinic for the treatment of this disease. And what we found was that the beta blockers by themselves were able to slow down the growth of the neuroblastoma tumors in mice, and it was increasing the regression that was induced by vincristine and delaying the regrowth following the vincristine treatment. This was associated with an increase in the anti-angiogenic effect of the chemo drugs in vivo and also an increase in survival. So I presented this data, not at the last meeting, the one before, in Haifa, and when Shripad Banavelli saw this data, he came to me and said, can you do something in angiosarcoma? Because I think it would be quite useful to use beta blockers in angiosarcoma. So for those of you who don't know angiosarcoma, it's a very aggressive form of vascular tumors. So it's basically the malignant side of hemangiomas. It's very rare. It's about 2% of soft tissue sarcomas. But when it's metastatic at diagnostic, the prognostic is very dismal. So what I did is that I used a model of bone marrow-derived endothelial cells that were either immortalized or transformed with the RAS oncogene. And I looked for a potential synergy between propranolol and the drugs that are used in the clinic for the treatment of this disease. So with doxorubicin, we didn't see much. With paclitaxel, we didn't see much. But with vinblastine, there was an increase in sensitivity. So we used the Shu and Talale method to calculate combination index. And we found that doxorubicin and paclitaxel were interacting through additivity or slight antagonism with beta blockers. But there was a strong synergy with vinblastine. So I was able to confirm that using a 3D model of tumor spheroids, where when we use a concentration of vinblastine or propranolol, which decreases the growth of those spheroids by about 20% when used alone, when we combine them together, we completely blocked the progression of those spheroids. So I told Shripad the way to go is to use vinblastine in combination with propranolol, and so he started to treat patients with this protocol here in the Tata Memorial Hospital. So up to this point, there's about 15 patients that have been treated, and there will be a poster tomorrow and the day after that you might want to see. I just wanted to highlight one of the case report we had, and that's the moment I think Shripad and I really started to think there's something there and it's really working. So this patient, a 72-year-old man, came into the Tata Memorial Hospital with a multifocal angiosarcoma with a massive tumor here in the periorbital region, as well as multiple lesions on the scalp, what you can see on the CT scan. And so this patient was offered a treatment with metronomic chemotherapy based on vinblastine and methotrexate in combination with propranolol. And this is the patient after 3 months, had a complete clinical response, and this is him after a year. So we published this data very recently, just last month, in eBiomedicine. So this patient I mentioned is patient number 2 here. He went on to live 30 months after diagnostic, which is extremely long for this pathology because the prognosis is much lower than this. So we included 7 patients here that we had enough follow-up on, and basically every single one of them responded, either partially or some of them responding completely. And we had like a median progression-free and overall survival of 11 months and 16 months, which is much longer than was ever reported in the literature. So based on this work that we've been doing, and the work of others, obviously, there's a lot of clinical trials now going on. Yesterday I checked on clinicaltrial.gov, and there's 41 studies that are referenced there in breast cancer, ovarian cancer, cervical, and so on. But we also have our own trials that are starting now. So we have a trial starting in France, which we call PROPAN-1 in angiosarcoma, where we're going to use different doses of propranolol with metronomic cyclophosphamide, and which it's led by Sébastien Salas. And we have another study which is led by Nicolas André, which will use propranolol at a fixed dose, but escalating doses of metronomic oral vinorelbine for children with relapsing or refractory solid tumors. And I highlighted here the funding bodies because Pan didn't really mention that, but it's sometimes really hard to fund these studies because most of those drugs, they are off patent. So it's hard to find a company that will sponsor the trial. So we're very thankful to Anti-Cancer Drug, which has been supportive of this work. And also recently we got funding for both of these studies from the French National Cancer Institute, which is a strong message for us that we're doing something that might be interesting. Okay, so now I'm moving on to the second part of my talk, which is called Next Generation Drug Propositioning. So as you could understand, my main goal is to develop better treatments for cancer patients. And the way people have been doing that for the last 20 or 30 years is to start from the omics, so whole genome sequencing, exome sequencing, SNP analysis from patient samples in order to identify novel therapeutic targets. Then you move on to high-throughput drug screening of diversity libraries to identify either a specific inhibitor or activator of these new targets, and then starts the long road of the medicinal chemistry so that you can develop a new drug. So all the meta-analysis of studies that have been done for the past 30 years have shown that it takes between 17 to 23 years to go from the target identification to the drug approval. So like Pan mentioned, there's literally hundreds, even thousands of drugs out there that are already available. We already know the PK data, the toxicity profile, and we also have high-throughput screening facilities. So I thought, is there a way we could do the reverse cycle and use drugs that are already available so we can identify new therapeutic targets? And I came up with this workflow here. So the first thing to do is to do high-throughput drug screening using libraries of drugs that are already available. And I use an approach which is called synthetic lethality. The idea is to screen the drugs alone and in combination with the standard of care for a given disease. So we can find drugs that will be active by themselves, but also drugs that can increase the efficacy of the current treatment. Once we do that, we identify the active molecules. And again, Pan mentioned that when we do a screen like that, we get 100, 150 drugs. So which one do we prioritize? Where do we go from there? And the idea that I had was to use the fact that we already know most of the molecular targets of these drugs because they've been studied extensively and there's a patent over them. So using a very simple bioinformatic analysis, I'm not a bioinformatician, so it has to be simple for me, we can identify all the molecular targets of these hits. And then I use a second screen, this time with siRNA, still with the synthetic lethality approach, so I can validate which one of those molecular targets are really important in the disease. So then from then, I can follow on my functional study by RNA interference to define the pathophysiological role of the key genes. I can also look at patient samples to define their prognostic and predictive value. And most importantly, I can use this data to select the drugs that are the most interesting. I'm going to select the drugs that are targeting those key genes. And hopefully, with this, we can develop new treatments and at the same time come up with the associated biomarkers. So we can go on to do a clinical trials selecting patients based on these biomarkers. So I've used this approach in different cancers, but what I'm going to talk to you about today is mostly on glioblastoma. So glioblastoma multiforme is a very aggressive and invasive form of brain cancer. It represents about 20% of all intracranial tumors. And the median survival is 15 months, and the 5-year survival rate is below 5%. So it's needless to say that we need better treatment for these patients. So I started by screening 3 different libraries, the Prestwick, the LOPAC, and the TOCRIS, which contains about 3,700 drugs and covers most of the drugs that are approved for clinical use in the US, in Europe, and in Japan. And there's also a lot of experimental drugs which are already in clinical development in those libraries. And I screened them, like I said, by synthetic lethality, so alone or in combination with the standard of care for glioblastoma, which is temozolomide, and also with EGFR inhibitors, because 50% of glioblastoma present EGFR mutation, but the clinical trials with EGFR inhibitors have been a complete failure. So I thought maybe in those drugs we can find drugs that can increase the efficacy of the EGFR inhibitor in this context. So using this approach, I've identified 201 drugs that I call GBM killers, so drugs that are going to be able to kill the glioblastoma cells by themselves, and also drugs that can enhance the activity of temozolomide or erlotinib. So then we did a validation screen, taking the top 280 of those drugs, and this time we used additional glioblastoma cell lines, still with the same setup. So using this approach, I was able to validate 81 of the GBM killers and 42 of the enhancers. So now we have 123 interesting drugs. So obviously, I'm not going to run a preclinical trial with 123 different drugs. I need to select the ones that are the most promising. So if we have a quick look at the type of drug we identified in the GBM killer category, not surprisingly, we have a lot of chemo drugs. You put chemo drugs on cancer cells in vitro, it's going to kill the cancer cells. A lot of experimental drugs as well, but also more surprisingly, drugs that are used in the clinic for very different applications. So anti-emetic, antibiotics, cardiac glycosides, statins, and so on. So what I did is that I did this bioinformatic analysis that I told you about. So I used different databases, but I just wanted to highlight the two that I think are the most interesting. There's a database which is freely available online, which is called DrugServe. You just type the name of your drug here, and it's going to list all the known molecular targets for this given drug. And I also use another one, which is commercially available, and it's an Indian company, so I wanted to highlight it. It's called GoStar. And it's even more exhaustive because it also includes data from docking analysis and also all the data from the patent. So sometimes you find more molecular targets in this database than in this one. There's a lot of overlap between the two. So using those two databases, I was able to identify a total of 1,257 molecular targets for those 123 drugs. So if you remember, I'm doing this to simplify things. And it's get even more complicated because now we have, 1,257 targets. So which one are we going to pick? So what I decided to do is to design an siRNA library to knock down each of those genes. But we didn't have enough money to do the 1,257. So we had to come up with an arbitrary cutoff. And we decided to focus on the targets, the genes that were targeted by at least 3 of the pharmacological hits. The idea was, if a gene is targeted by several of the drugs that came out of our screen, it is more likely to have an importance in the disease than if it's targeted by just one. So I represented them here, and obviously I'm not going to go through the 388. But what I wanted to highlight is the fact that some of those genes are well-known targets for improving therapy, like the P-glycoprotein or some of the cytochromes, but it might, in fact, impact toxicity as well. What was more surprising is to find a lot of neurotransmitter receptors, like the adrenergic receptors, the muscarinic receptors, dopamine receptors, and then we have a lot of classical therapeutic targets in cancer, like mTOR, MAP kinase, and so on. So I developed, I designed an siRNA library with 3 individual siRNA sequences to target each of those 388 genes, and I screened them in 4 different GBM cell lines, again, using a synthetic lethality approach, so alone, or in presence of temozolomide and EGFR inhibitor. So I just wanted to show you a little bit what the data looks like. So at the end of the treatment, we stain the nucleus of the cells with USHT, and then we take pictures of each of our conditions. So we had a negative control siRNA and a positive control siRNA, and this is 2 of our top hits, polo-like kinase 1 and R1, which is a ribonuclease. And what you can see is that when you knock down those 2 genes, there's way less cells than in the negative control, and also it sensitizes the cells to temozolomide. So I'm showing you 8 pictures here. There was 64,000 pictures that were taken in this project. And so obviously I didn't quantify everything by hand, and we did all that using softwares and computers. And so when we did that, the analysis allowed us to identify 219 key genes. So we have genes that will be important for the regulation of proliferation in GBM cells, which are coming from the screening in the absence of drugs, and also genes that are important for regulating the response to temozolomide or erlotinib. So we have 98 genes that we can now target that are involved in temozolomide resistance. We had 156 genes that are important in EGFR inhibitors. But for now, what we're focusing on are those 38 genes that were in the center of the drawing before, which are involved both in proliferation of GBM cells and in drug resistance. So I used the color code here to highlight some of the results we got. Every gene that's represented in blue here are genes that were discovered in the past 5 years as being new targets in glioblastoma. So we have the Aurora kinase, the polo-like kinase, check 1. And so it was quite interesting to see with this approach, we rediscovered things that were discovered 5 years ago, or some of them just last year. In green are genes that I consider to be classical therapeutic targets, like AKT1 or genes involved in the cell cycle or of the tubulins. And in red are all the genes that if you type the name of this gene and glioma next to it, nothing comes up. It was never reported as playing a role in glioblastoma. So also it highlights the strength of this approach, that you can identify a lot of new potential therapeutic targets in a given disease. So now we're starting the functional analysis of all of this. So we're studying their gene expression level in different glioblastoma cell lines, as well as glioblastoma stem cells. And what we found by using this approach was that there was a lot of variation from one cell line to another, but we also found that some of those genes were either downregulated in glioblastoma stem cells, like the polo-like kinase 1 or the aurora kinase A, and some of them were overexpressed in glioblastoma stem cells, and we were able to confirm that when antibodies were available by Western blotting. So in terms of fundamental aspect of this work, we can generate a lot of new knowledge. And so we are in the process of validating all this data using, again, the functional studies. So for instance, with our top gene here, we get a 95% knockdown with the different siRNA we used in the screen, and it was a complete knockdown at the protein level. And you can see that we were able to confirm that if you knock down this gene, you completely abrogate the growth of the tumor spheroids. But the main reason I'm doing this is to select my drugs. So now I can go back to these key genes and select the drugs that are going to target them. So we have here disulfiram, for instance, which is able to target PLK1, aurora kinase A, and 885. And this is quite interesting because there's 3 clinical trials at the moment using disulfiram in the treatment of glioblastoma. And we have other genes as well, other drugs that we can use that look very interesting. So again, I needed to come up with some kind of analytical tool to be able to pick the drugs that we wanted to use further. So what I did is that I gave each of these drugs what I call an siRNA score, which is based on the molecular targets and the effect of the knockdown of these molecular targets in the cells. So then I was able to classify all of the drugs that came out of our screen. So there was a lot of experimental drugs in this, and we decided to focus solely for the moment on drugs that are already approved. So you have sunitinib that comes up really high, disulfiram, apomorphine, and lovastatin. So now we're in the process of testing each of those 20 or 22 drugs in the different cell lines, either in alone like this, or in combination with temozolomide, and also in combination with themselves. So it's a little bit tricky, but what we found was a lot of the drugs that are used for ementic infection are very, very active. We have the disulfiram as well, which is used for curing alcoholism, which is also very active, and some others. So if we go back to my workflow, at the moment we're here. So we've identified all the key genes, and we've started the functional study. We're looking at patient samples, whether any of these genes are providing some prognostic or predictive information, and we're also starting the preclinical efficacy. So in 2 years, I don't know where we're going to be for the next Metronomics meeting, but I'm hoping I'll be able to show you all the finalized data for this project. So in conclusion, what I call next generation drug repositioning is this idea of using high throughput pharmacology screening with functional genomics using bioinformatics tools in the middle. That it's all great and good what we're doing with cell lines, but what I ultimately want to do is to treat real tumors in real patients. So how are we going to do this with this kind of approach? The idea is that with this work, we're generating a lot of data. So we're using different cell lines in which we're screening a lot of different drugs. Then we're doing siRNA screening all those cell lines, and we can do the genomic profiling of each of the cell lines we've used. So we can then use what we call machine learning or artificial intelligence, whatever you name it, to develop a predictive algorithm based on this data, so that we can say, based on the genomic profile of a tumor, that this drug and this drug are the most promising one to use in clinical trials. And so the idea would be to validate that using patient-derived xenograft to validate the algorithm. So to finish, I'd just like to acknowledge the people that were involved in this work. So this was my group when I was back in Sydney. And so the people that were involved in the beta blockers work a lot. And now back in Marseille, so working a lot with Nicolas André and Manon Carré, and also with my students. I'd like to thank Shripad Banavali, obviously, and also Guillaume Pina, who did the sRNA screen for me at the platform. And I'll thank you all for your attention.